Hello guys, this is my first video and I am starting from embryology of eye because this is a very basic thing that all should know, especially optometry students. So during the third week of pregnancy, germs layer start developing. As we know, there are three germs layer through which our body parts developed. So these are ectoderm, mesoderm and endoderm. Ectoderm is the outer layer, mesoderm is the middle layer and endoderm is the inner layer. Mostly the eye develops from ectoderm layer. It also develops from mesoderm but mostly develops from ectoderm. There are two parts of ectoderm, neuroectoderm and surface ectoderm. There are three germ layer. Ectoderm eyes develop from ectoderm having two layer, neuroectoderm and surface ectoderm. Now, neuroectoderm develops into neural folds or neural groups which then developed into neural tube. Means first neural ectoderm which developed into neural folds or it is also called neural groups. Then it develops into neural tube. This neural tube helps in the development of brain or central nervous system. As we know, there are three parts of our brain: forebrain, midbrain and hindbrain. Forebrain is called prosencephalon, midbrain is called mesencephalon and hindbrain is called telencephalon. Now, prosencephalon helps in the development of eye. Eye develops through prosencephalon. So, eye develops after 22 days of embryo development. Embryo after 22 days of embryo development, eye develops. That time it is about 2 mm. It is important. It is of 2 mm. When we do the cross section of prosencephalon, we get a neural plate. It is a prosencephalon, it is a outer patching, and when we do a cross section we get a neural plate. Now step by step we will understand. So it is a neuroectoderm. So the thickening of the neural plate occurs called placode. We know when we do the cross section of the prosencephalon we get neural plate. Then thickening of the neural plate occurs which is called placode. Then in step 2 the placode or the neural plate get depressed. When it get depressed it is called invaginate. Step 3 the invaginate or the neural plate get more depressed. It is called optic sulcus. Then in step 4 the optic sulcus or the placode or the neural plate which get depressed more get elongated. Here you can see in my diagram it is elongated more. So it is called optic vesicle. It is called optic vesicle. Now the proximal part means here the proximal part, this proximal part, it get elongated and constricted. This get elongated and constricted which is called optic stalk. Optic stalk. See, the proximal part of the neural plate get constricted and elongated which is called optic stalk, like this in this diagram. And the depressed part, it is the depression part and it become round, like you can see in this diagram. It become optic stalk and it become round. Now in step 6, this is the neuroectoderm. As we know, there are two parts of ectoderm, neuroectoderm and surface ectoderm. This is neuroectoderm and now this is the surface ectoderm. Then what is the role of surface ectoderm? See, this surface ectoderm then become thick, like we study in step 1, it become thick. Now this become thick. When this become thick, it is called lens placode because through this lens formation occurs. So we called it lens placode. Now in step 7, this lens placode get depressed. It called invaginate. As we see that the neural neural plate get depressed, we call it invaginate. So in this also we call it invaginate. Now Step 8, this become more depressed and it enters inside the optic curve. It is an optic curve. It enter inside the optic curve. It is called lens pit. What it is called? It is called lens pit. Then there are two walls of optic curve, inner wall and outer wall. I will tell this later. So the depressed part or the optic lens pit get depressed into the optic curve, means it get detached from the surface ectoderm. See, in step 9 it get detached from the surface ectoderm. Now in step 10, this lens vesicle is covered from the superior and lateral quadrant of the optic curve. Later it get covered from the inferior quadrant. This process of covering of the lens vesicle through the optic curve from lateral and superior quadrant and later through the inferior quadrant is called choroidal fissure or fatal fissure. Then if due to any condition this is not covered, it is called coloboma. As we know, if anything is not covered or not present, the tissue is absent, then it is called coloboma. There are two types of coloboma, typical coloboma and atypical coloboma. When it will be typical coloboma, it is lens vesicle is not covered through inferior quadrant, and it is when it is atypical coloboma, it is not covered from lateral and superior quadrant. For this class I will stop here. You can also follow my notes for long question answer and everything I have written in details, so you can follow it. Thank you.